Hello, and thank you for this opportunity for me to offer you this history of Dent disease, an entity that really wasn't recognized as a discrete disease until the mid-1990s. And so it is wonderful to reflect on the major advances that have occurred during that period of time. I regret that the time allotted for this talk today is brief and therefore will not allow me to acknowledge everyone who made significant contributions to our understanding of Dent disease. But with that disclaimer, we can move on to the next slide, which summarizes what we all recognize as the classic features of Dent disease: a proximal tubulopathy with hypercalciuria, stones and nephrocalcinosis in many of the patients, renal failure in some of the patients, and most variably rickets, with the defining features being low molecular weight proteinuria and hypercalciuria, which represented the clinical criteria for entry into almost all of the studies that I will be describing today. And if we use this as the defining characteristics, the next slide lists a number of reports that were offered before we recognized and labeled this as a single entity, going back to the 1964 report by Dent and Friedman. But even before that, in 1962, René Habib and colleagues in Paris reported a family with hypercalciuric rickets, proteinuria, stones, and renal failure, very strongly suggestive of Dent disease. Unfortunately, when I reached out to René Habib for samples back 25 years ago, it was already too late for her to identify and contact the patients, and so we will never have definitive confirmation of that suspicion. And of course, we all look back to 1964 and have named the disease in honor of Charles Dent, who reported these boys with hypercalciuric rickets and renal tubular damage. And we owe a huge debt of gratitude to his junior colleague, Oliver Wrong, pictured to our right of Charles Dent, moving clockwise through the photos to the right. Oliver followed Charles Dent's patients and many other patients with Dent disease over decades, characterized them fully, and we owe a great debt to him for his contributions to our understanding of the phenotype of Dent disease. In 1974, Vardaman Buckaloo in North Carolina, who's the distinguished-looking gentleman in the sweater pictured below Oliver Wrong, reported a family with what he described as hereditary RTA with hypercalciuria. Years later, when it was confirmed that they had mutations in CLCN5, he acknowledged in print that his description that it was hereditary RTA was incorrect, that the acidification defect was in fact secondary, most likely, to the nephrocalcinosis and not a primary feature of the disease. And so it was clear that they had Dent disease. We've also learned a great deal from the description of large numbers of Japanese children with hypercalciuria, nephrocalcinosis, and low molecular weight proteinuria reported by Takashi Igarashi, pictured at the bottom. But the key link to being able to identify the gene came with a description of a family by Paul Freimoyer and colleagues. Paul is pictured in the white coat on the middle left. And it was this family that allowed linkage studies to be done. The next slide shows the key member of that family on the right with her 4 children, 2 of whom have clinical Dent disease. And it was her concern for their future that prompted her to gather the family information and work with us to put together a pedigree demonstrated on the next slide, with 162 members of this family going back 6 generations to the late 19th century when they arrived in northern New York State from Liverpool. And it is very clear on this pedigree that the inheritance is X-linked recessive. And so with that information, I and colleagues collected blood samples on 102 surviving members of this family including a 2-month-old child and a 100-year-old woman and everybody else in between. And went to Raj Thacker's lab, where I learned to do restriction fragment length polymorphisms. And one branch of that family, including the woman I just showed you, is illustrated on this slide. Studied with the RFLP probe M27 beta on the X chromosome. And as you can see on this portion of the pedigree, the affected males in the solid squares all demonstrate a 7.5 kilobase allele for this probe. The unaffected males in the open squares all demonstrate a different allele, and the carrier females have the 7.5 kb allele plus one other allele consistent with their having two X chromosomes. This held true across the entire pedigree, and so it allowed us to demonstrate with a high level of confidence, a log score of almost 6, that the location of the gene, next slide, was on the centromeric region of the short arm of the X chromosome. Simply by localizing it, we were able to demonstrate that it was distinct from other X-linked renal diseases such as hypophosphatemic rickets, Alport syndrome, nephrogenic diabetes insipidus, and the Lowe syndrome. In fact, we were not surprised that it was separate from the Lowe syndrome. We had ruled that out in this family through the absence of eye disease or mental developmental disorders. But with this information, Raj Thacker went back to his shelf of samples that Oliver Wrong had collected over the years, including this family of a patient originally followed by Charles Dent, not reported by him. And the affected males once again are indicated with the filled squares. And as you can see, for the probe M27 beta, there is no allele for any in the lane underneath any of the affected males. And it was not a technical problem because using another probe on that same samples showed an allele for everybody. This was strongly suggestive of a deletion of this region, and so Raj and his team saturated that region with markers and demonstrated that there was a deletion of 512 kb, which contained only one potential gene, which we now know as CLCN5. Next slide shows the CLC family of chloride transporters, and we owe a great debt to Thomas Jentsch, an electrophysiologist in Hamburg, Germany, who had described the first voltage-gated chloride channel in the giant squid axon, and then by homology cloning was able to clone the major chloride channel in human muscle, CLC1, which is mutated in the most common form of myotonic dystrophy, congenital myotonia. The members of this family, as indicated on this dendrogram, in the lower portion of the dendrogram are all chloride channels, whereas those in the upper half of this dendrogram are antiporters, proton chloride electrogenic antiporters, and that includes CLC5, responsible for Dent disease. Next slide shows Raj Thacker again, who gathered samples from a range of patients and demonstrated 11 different mutations in patients, including ours and those from Canada and Italy and elsewhere, and demonstrated that despite differences in reported phenotypes, they all had a single molecular cause, mutation in CLCN5, and these mutations ranged from truncating mutations and non-truncating mutations, including missense and nonsense and frame shift. And the next slide, years later, reported in 2023 by John Liskey and his group, using samples generated through the Rare Kidney Stone Consortium, the Dent Disease Registry, were able to demonstrate, consistent with the literature in this large sample of over 100 patients, that a third of them the most common group, had missense mutations, but there were a range of nonsense, frame shift, and other mutations as well. And so here we see on this slide a summary of the missense mutations, most of which are bunched in the transmembrane domain of the protein, including the mutation in case 1 of Charles Dent. Charles Dent reported 2 patients, and his second patient had no mutation in CLCN5. So stay tuned on that one. And so, as you might imagine, the knowledge that this gene was disease-causing in humans focused a lot of attention by many laboratories, not least of which was Thomas Jentsch's laboratory, who demonstrated that, contrary to earlier suspicions, this was not a chloride channel, but in fact an electrogenic chloride proton cotransporter that could transport chloride channels in either direction depending on the driving force. We owe a great deal of our understanding of this transporter to Thomas. But we've also learned a great deal about its larger function in renal physiology from people like Olivier de Vuist, a Belgian scientist who is now in Switzerland, who's contributed a great deal to our understanding of the role of CLC5 in early endosomal function, in membrane recycling, in protein trafficking, and protein processing, and in general in proximal tubular function. We are also grateful to both Thomas Jentsch and Bill Gugino at Johns Hopkins, whose laboratories independently generated knockout mice of CLC5, fulfilling Koch's postulates by demonstrating that knocking this gene out in mice reproduced virtually all of the features of the human disease. It also provided a valuable animal model for studying the role of CLC5. And so we now recognize Dent disease type 1 as those patients characterized by mutations in CLC5, but we know that this only accounts for about 60% of patients who meet clinical criteria for the phenotype of Dent disease. And over the years, my laboratory collected samples sent to us from across the world of families who met these criteria for Dent disease, and when we found mutations in CLC5, we would collect them and report them. But a number of these patients, a dozen that we had collected, a dozen families, did not have mutations in CLC5. And one of those families, pictured here, was a multi-generational family from Hungary sent to us by János Mátyus in Debrecen, Hungary, in whom the inheritance pattern is consistent with X-linked recessive inheritance, but when we used markers linked with the region of the CLCN5 gene, we ruled out linkage to that region, and for good measure, we sequenced these patients and demonstrated that their CLCN5 sequence was normal. And so this family was large enough for us to do mapping, and on the next slide, we see the results of that mapping, which narrowed the region to a relatively small region on the long arm of the X chromosome that contained about a half dozen genes, including OCRL1, but we did not initially sequence OCRL1 because we had, as entry criteria for this study, ruled out patients who had oculocerebrorenal, had eye or brain abnormalities. And so we focused on the NHE3 sodium proton exchanger, which is also in this region. That sequence was normal. And so then we did sequence OCRL1 and in fact demonstrated mutation in this family as well as several other families, and so reported, next slide, what we now know of as Dent disease type 2. Patients whose phenotype of Dent disease, renal phenotype, was explained by mutations in OCRL1. It's the same renal phenotype as patients with the oculocerebrorenal syndrome of Lowe, the syndrome that gave the OCRL gene its name, but these patients lack the eye and brain abnormalities. And Robert Nussbaum is the man who cloned the OCRL gene, and to whom we all owe our much of our knowledge of OCRL to him, and he collaborated with us in this study. And so on this slide, we see a schematic of the OCRL gene, exons 1 through 23, with mutations in the full Lowe phenotype at the above that schematic. And we see it's a range of mutations, including missense, deletion, nonsense, frameshift, and splice site mutations. And the mutations in patients with Dent2 below, including our original report in 2005 and our subsequent report labeled current, which included Charles Dent's patient number 2 with a frameshift mutation in exon 7. And so we were able to demonstrate that all of the truncating mutations in patients with Dent disease occurred in exon 7 or earlier, whereas that's not the case for the patients with Lowe syndrome. And since we found a methionine transcription initiation site early in exon 8, we were able to offer an explanation that truncation before exon 7 still allows for transcription of a gene representing the transcripts 8 through 23, which include the full functional domains of the phosphatase, the ASH, and the Rho gap domains. There are also missense mutations in Dent2 patients in the phosphatase domain, but these missense mutations are functionally less severe than the missense mutations in patients with Lowe's. So we have at least a partial genotype-phenotype explanation for Dent2. And so, as we've seen, Charles Dent's 2 patients included his case 1 with a mutation in CLC5 and his case 2 that we were able to demonstrate had a mutation in OCRL1. And Oliver Wrong, over the years, kept saying that case 2, who did not have mutation in CLC5, had something funny going on because he was mentally backward. He was not severely disabled mentally, as many patients with Lowe syndrome are, but he did have an IQ of only 65. And Oliver made a point of this, and he was absolutely right, since we now know that mental developmental delay probably was a partial phenotype from OCRL1. And so there's much more that we know, but I've not been able to summarize. And this history is still being written. We look forward to a deeper understanding of the physiology and the pathophysiology accounted for by these 2 genes, particularly the factors contributing to renal failure and the other features of Dent disease, and understanding the phenotypic heterogeneity. Our understanding of renal failure may be enlightened by the description by that report from John Leesky's group that almost all of the patients with renal failure had truncating and not missense mutations. A potentially important clue. We also look forward to understanding the genes responsible for the remaining 25% of cases, which presumably, since many of them are inherited, presumably also are explained by other genes. And most exciting will be our understanding of opportunities for therapies. And it's exciting to observe that there are already promising early reports of therapies that, should they fully play out, might make a difference for patients with Dent disease. And so with that, I'll conclude with acknowledging all of the many people who will contribute to the history of Dent disease yet to be written. And that includes some of the people in this slide, those of us who gathered for the Kidney Stone Research Day at the Mayo Clinic last October with John Leesky in the center, and several others in this picture who are actively working on dent disease, including therapies for dent disease. And that includes people not pictured here from Spain and France and Italy and elsewhere across the world who are actively working on this now. And so with that, I thank you very much for your attention. Have a good day.